Okay, in the previous lessons, we have talked about these R3 already, okay, which consists of 3 layers. The innermost layer is called tunica intima, consists of the endothelium, okay, consists of endothelium, okay, and then this endothelium actually stick or actually okay, stick of this basement membranes, okay, stick on basement membranes, and then the second layer, thicker layer called tunica media, okay, consists of the smooth muscle, okay, consists of smooth muscle and a lot of this elastic fiber and some extent to means we have this elastic, sorry, the elastic fiber, okay, sorry, collagen fiber, okay. So now we talk about this, the outermost layer, we do have the tunica externa, mostly will be connective tissues and also the collagen fiber. So let us look at the vein, how the vein actually different from the artery. Okay, so first of all, we do know that as blood enter into the capillary beds, okay, then do exchange already, then they start to join back, okay, these join back. So first they join back and form venules, okay, join back and form venules, and these venules will going to join together to form the vein. So the largest vein that we do have is termed as the vena cava. Okay, so vena cava will bring the blood enter into the heart. Okay, so vein, when I use the word vein, vein basically return blood to the heart. So we do have a lot of vein, and basically or generally, okay, all the vein except pulmonary veins, they are deoxygenated. Are you clear? Okay, the ones that oxygenated one is the, okay, pulmonary vein. So but when we talk about the word vein, okay, when you use the word vein, vein generally means that they must return the blood to the heart. So it doesn't care whether they are oxygenated, the blood that carry are oxygenated or deoxygenated. Okay, so again, the wall also consists of 3 layers. They are tunica intima, tunica media, and tunica externa. So same thing, you look at this tunica intima consists of what? Still the same, endothelium. Okay, so low resistance. So tunica media, because we do know that the blood pressure, okay, or the pressure of the blood inside the vein is quite low, okay, it's negligible in some of the vein. So therefore, we do not need to have a very thick tunica media, okay, if you compare, no need to be so thick. So again, they do have the muscle, okay, so we have the smooth muscle, okay, and elastic fiber together with collagen. But elastic fiber, the quantity is quite low. Are you clear? Fewer elastic fiber and muscle, not fiber, sorry, no. Okay, so the tunica media of the vein is much thinner and has far fewer elastic fiber if you compare to the artery. Okay, tunica externa almost the same as, okay, we're comparable, artery and the vein comparable. Then you can see that actually tunica externa almost the same for both vein and also the artery. So both of them also consist of connective tissues with a lot of collagen fiber. Okay, a lot of collagen fiber in this tunica externa. Okay, so collagen fiber basically give the shape and also it prevents sorry, not prevent, basically I mean allows our, it connects, okay, this collagen fiber connects the vein to the surrounding. Okay, if we don't want, I mean our vein actually move here and there. Okay, so in terms of wall to lumen ratio, okay, lower. So means that if you look at this, the wall to lumen, so wall actually thinner, lumen wider. Are you clear? So therefore, the ratio of wall to lumens, okay, wall to lumen ratio actually will be low. Are you clear? And I did tell you guys that wall to lumen ratio is not a totally everyone agree to use the same thing, wall to lumen ratio. Some may use lumen to wall ratio. It doesn't matter. Are you clear? okay, as long as you use the correct, if in this case, lumen to wall ratio will be very high, okay? So by the times, as I say that the blood enter into the vein, the pressure have dropped to very, very low value. So it means that we do not need to have a very thick wall for the vein, okay, in general. So now the pressure is so low, how we can actually, okay, move the blood along this vein. So now we have to know that, okay, in terms of the comparison between, okay, as I say that if you compare between this artery and also the vein, they are the same types. For example, you compare the hepatic, okay, artery and hepatic vein, renal artery, renal vein, okay, then it's comparable. You don't compare the vein, vena cava with artery, then difference. Are you clear? Vena cava, the wall is much more thicker, okay? So if you compare, you can see the wall to lumen ratio, okay? You can see that the red, dark red color, the dark, can you see that, this one? Okay, those are the tunica media. Okay, tunica media. So this lumen, and you can see the outside layer, they are tunica externa. Can you see that? Okay, a lot of connective tissues. Okay, so if you compare both of them, so you can see that tunica externa relatively the same. Are you clear? But in artery, you still have some elastic fiber. Can I see that elastic fiber? But in the vein, no elastic fiber. Most of are collagen fiber. In terms of number of smooth muscle, can you see that? In the artery, in the muscular artery, you can see that they have a lot of smooth muscle. Here, lack of smooth muscle. Okay. Now, one thing about if you compare between the artery and the vein, look at the tunica intima. Tunica intima for the vein, relatively smooth. Sorry, relatively smooth in this case. But this one, can you see that? We call it crinkled. This structure, tunica intima, okay, this structure is called crinkled. Means that folded, okay? Now, don't always think that folded must be, okay? Don't always think that folded must be increase the surface area. You increase surface area for what? No exchange is going to be done in the artery. Correct or not? So why crinkled in this case? Why crinkled? Basically, this crinkled allowed the wall to be stretched. Okay, what's this mean to be stretched in this case? Now you try to imagine, if I have a one piece of paper, okay, you have one piece of paper like this, you pull it from both sides, you're going to tear the paper. Correct or not? Okay, but if the paper is something like this, okay, the paper, so if you pull it, then you will be able to expand it. Correct or not? Can you see that? You'll be able to expand it, make it flat. Are you clear? It won't tear. So this crinkle allowed the wall to be stretched and then prevents this endothelium, okay, from the tear, okay, from damage. Okay, you won't damage this endothelium. Are you clear? So always remember, no gas exchange, no nutrient waste exchange in the artery. So this crinkle is one of the features of the artery so that you won't tear or damage the endothelium when the wall is stretched. Are you clear? Okay. So we do know that the wall thinner because we do not need to actually bear with for vein, they do not need to bear with the high pressure. Okay, that means the blood. So therefore, if no pressure, how the movements of the blood flow happen inside the vein. So the blood flow actually depends on the skeletal muscle, the contraction of skeletal muscle and the valve. Okay, don't say muscle only. Which kind of muscle? Because muscle, we have a few types of muscle. We have smooth muscle, cardiac muscle, and also skeletal muscle. So in this case, it's a skeletal muscle. Okay, and also the valve. So it means that if you look at these diagrams here, the very, very prominent features of the vein, they have this valve known as semi-lunar valve. Okay, this valve is semi-lunar valve. So this semi-lunar valve will ensure that the blood only moves in the correct direction, means forward direction, won't have the backward, backflow of the blood. So basically, this valve divides our vein into few compartments. So you see the compartment number 1, compartment number 2, compartment number 3. It becomes something like a compartment, a segment. Okay. So when we move, what will happen? When we move, our muscle contract. So when muscle contract will squeeze the blood from one region to another region. But when the muscle relax, this blood cannot backflow. Won't be able to have the backflow of the blood due to what? Due to the presence of the semilunar valve. So this semilunar valve will close. Are you clear? Will close. Therefore, you won't be able to have the backflow of the blood. This is how the blood flow take place inside our muscle. So this is something like a door. Okay, you can imagine that. So, if you have something like this, okay, this is the pillow, and I have a door. So this is the what we call this the fix of the door. Okay, so means that if I go through this way, no problem. I just need to push it. Can you see that? So the pressure of the blood, because you squeeze it, right? You squeeze, so the blood will push open this valve. It's not wall, the valve. Are you clear? But if you want to push it from this way, impossible. Clear not? Because you have something to stop the wall, as the door from opening another side. Are you clear? Okay, so means that you don't think that the blood is clever enough to actually open it. I mean, like pull the door. Okay, it will only push because of pressure. So therefore, you can see that this pressure, I mean the pressure exerted by the muscle, make sure that our blood move in the correct directions. Are you clear? Any questions so far? Okay. So we've done for the veins. Then we come to the connections between the artery and the veins, or more precisely, arterial and venule, which are capillaries. Okay, so this capillary, you won't see them as one capillary only, but they form a network, and this network is called capillary bed or capillary network. And very important, we want to increase the total surface area to volume ratio. Okay, and you look at the capillary compared to the artery and vein, they're relatively, the wall are very, very thin. Okay, and consists of one layer of the cells, and this layer of the cells highlights is endothelial cells only. Are you clear? Or endothelium. Now, what's the difference between endothelium and endothelial cells? We use the word endothelium, means tissues. So the cells that form the endothelium is known as endothelial cells or squamous epithelium. Are you clear? You can use either endothelial cells or squamous epithelial cells. Sorry. Are you clear? If you use the word epithelium, means that it's tissues. Always remember, okay? Endothelium, tissues. Epithelium, tissues. If you refer to the cells, then you have to put epithelial cells or endothelial cells. Are you clear? Okay. So they are the same thing, guys. They are same thing. Squamous epithelial and endothelial cells, they are the same thing. It's just that in the blood vessel, we give them a special name called endothelium, okay, or endothelium. Okay, so the capillary wall extremely thin. Okay, I know that most of the time students like to use one cell tape. No, yesterday or the previous lessons we have learned about it. We do have even one cells. It can be very thick. If let's say columnar epithelium, columnar epithelium is very thick. Cuboidal is very thick. Cannot. But for exchange to take place, capillary formed by endothelial cells, which is very, very thin cells, and the diameter of the capillary almost same as the red blood cell. In fact, red blood cell have to squeeze through the capillary. So why we need to do this? Okay, and the diameter of red blood cell is roughly about 7 micrometer. So by doing this, we were going to have a short diffusion distance. Are you clear? Okay, so let me draw the structures, then you can see the reason already. Okay, why we don't make it large in diameter. Now, let us look at this capillary. Capillary. So we do know that capillary formed by this flat squamous epithelium. So it will be something like this. Okay, you look at capillary in an A. So flat cells, and then okay, so second cell. Okay, third cells. Too thick. They are very, very thin only. And let's see, okay. So surrounding these cells will be the basement membrane. Okay, our basement membrane is very important to anchor, okay, to anchor your cells. If no, with the blood will flush away all the cells here. Are you clear? Okay, so this capillary, the structure of capillary, only one layer. So first thing, how they adapt to the function for capillary first? They are very thin. How thin? How thin? So it consists of one layer. Don't say one cell thick. One layer of endothelium or squamous epithelium. Okay, so it's so thin that the regions where you have nucleus, can you see that it show the protrusions in this case? Okay. Second, they have the lumens diameter. Okay, they have how to put this? Sorry, they're small lumen. Okay, they have small lumens. How small? So around 7 micrometer. Okay, can I see that? So it means that the red blood cells will be the size almost the same as the lumen. Can I see that? This red blood cell. What's so important about this? When the small lumens dense, both these features actually provide short diffusion distance. Okay, can I see that? It provide a very short diffusion distance. Therefore, we increase the rate. Rate of what? Rate of the gas exchange. CO2 move out from the cells, enter into the blood, and then the O2 from the blood enter into the cells. Also, to get the supply, increase the rate of the supply of nutrients, and also increase the rate of removal of waste. That's very important for the capillary. So what it means that short diffusion distance? You can see that if O2 actually at the center of the red blood cells, can you see that this is the diffusion distance? Can you see that? Okay, the diffusion distance. But if let's say you have a very large lumens, try imagine very large lumen of capillary. Okay, so it means that a red blood cell is something like this. So from the center of the red blood cell to the capillary, the wall, it will have a very, huge diffusions or long diffusion distance now. So this one will decrease the rates, which is what we don't want. Are you clear? So therefore, if we have a very small capillary, and this is instead of having one, we actually increase, right? Form capillary networks. Okay, so the third one here, they are highly branched. They're highly branched. Okay, in this case, forming capillary network. Okay, forming capillary network. So again, we increase. What we increase? We increase the surface area for exchange. Are you clear? We increase the surface area for the exchange. Here, so this one I remove it lah. Okay, before I take the photo. Okay, clear, guys? Any question? Oops, what I'm doing? Sorry. Wait, guys. Yep. Okay, the exchange including, that means the supply, nutrient, waste, gas exchange. Okay, so this is the capillary. Okay, you can take a photo. Okay, so you can see that this is the features of the cells in three-dimensional. Okay, three-dimensional. So you can see that this is the one's endothelial. Okay, one endothelial. They had the intercellular cleft allows the exchange of the material, but you can see that all these endothelial cells, okay, are anchored onto these structures called basement membranes. Okay, so we have endothelial cells. This is the nucleus of the endothelial cells, and we do have the basement membrane to make sure that you don't flush away, okay, the endothelial cells. Okay, so this is the structure of capillary. So in fact, we have three types of the capillary. Okay, three types of capillary. So first type capillary called continuous capillary. Second type where we're going to learn in the kidney. This one appear in the kidney. Glomerulus. Okay, we'll learn about this for ultrafiltrations. Then we have this fenestrated capillary, and the last one called sinusoidal capillaries. Okay, so you can see that in terms of the structure here, continuous, basically the basement membrane, you can see that no pore, basement membrane, no pore, and the cells don't have pore also. Can you see that? The cells don't have gap and don't have the pore. So this kind we call continuous capillary. Most of our cells, most of the blood capillary using this. But in the kidney, because we want to have the ultrafiltrations, so in order to have ultrafiltration, you can see that now our capillary cells, they are porous. Can you see that? So this pore is called as the fenestration. Okay, the pore is called as a fenestration. But you can see that now we still have this basement membrane. Can you see that? Continuous one. It's just that the cells have the pore. So this pore allows the ultrafiltration to take place. And we do have some sinusoids. Okay, this sinusoid capillary or sinusoidal capillary basically have this a lot of big gaps. So this one will increase the exchange even more. Are you clear? And you can see that we have the incomplete basement membrane also for the exchange. Okay, so for our syllabus, we only need to know these two, continuous and fenestrated. Okay, continuous and fenestrated. Continuous is a normal one, fenestrated found in the kidney. Are you clear? Okay, so we come to the last part for our class for the blood vessel. Now let's look at it. For the blood vessel here, very, very important when you compare, we take the wall to lumen ratio. Wall to lumen ratio. And you can see that it consists of many layers. So A must be artery and vein. So which one artery, which one vein? Question sometimes asks you to identify. Okay. So they are under the same magnification. Can you see that? Because take from the same photo. So same magnification. But A and B, they are not the same because capillary here are shown in the figure B. Okay, a higher magnification compared to artery and vein in A. Why? Because there are two different photos. Okay, so you can see that these are the simple squamous cells, simple squamous epithelial cells, or we call endothelials. You can see that the nucleus here, they are very, very thin only. So they are not same magnification, different. So much more higher for the capillary. Okay, so but here in A, how we differentiate between the artery and vein, we always check the lumen wall to lumen ratio. Can you see that thick wall, narrow lumens compared to thin wall, large lumens? Can you see that? So it means that this one must be highlight, must be vein. This one must be artery. Okay. So now we compare three of them. Okay, we compare three of them: artery, veins, and capillaries. So first, in terms of blood transportations, generally artery, okay, or all artery transport the blood away from the heart to other tissues. Okay, so we have aorta, so artery. So this artery is a muscular artery, okay, basically. Aorta is the elastic artery, so muscular artery, and then to arterial. So after that, arterial branch to capillary. So capillary actually they link the artery to vein. They serve as sites for material exchange. Okay, and then from the capillary joint to form venule. From venule joint to become the vein, and the vein joint all together become vena cava. Okay, in general. Okay, so what is the role of the vein? Transport the blood from other tissue towards the heart. So in terms of thickness of tunica media to lumens, okay, we will focus in tunica media only in this case. Then you can see that's high ratio, okay, due to the thick tunica media and low diameter of the lumens. Capillary does not have tunica media, so that's why they only have lumens. We cannot do the ratio here. But for the vein, they have a low ratio due to the thin tunica media and high diameter of lumens. Okay, semilunar valve, artery, absence. Okay, no semilunar valve in the artery. Capillary, okay, absent also. We do not have the semilunar valve. Okay, but vein, yes, semilunar valve present to prevent the backflow of the low pressure bloods. Okay, how about the blood pressure? So it's quite high. So that's why we need to have the elastic, high elastic fibers. Okay. In terms of the capillary, no pulse. So blood pressure is lower than the artery. And same goes to the vein, which have the lowest blood pressure. Okay. In terms of blood flow, very rapid because of the surge. Capillary will be slow. We cannot be fast. If fast, then we cannot have enough time to do the exchange. And the slowest actually take place inside the vein. Okay, it depends how slow. It depends on whether you move or not. If let's say you didn't move, the blood basically will stop moving. Okay, because no muscle contraction. Oxygen content, so oxygen rich except the pulmonary artery. So it's a mix of the oxygenated and deoxygenated. So it depends on which end. So if you look at this, if I draw it out, here's a capillary. So if this is the arterial end, this is a venule end. So this side are more to oxygenated. And this side will be deoxygenated. Okay, so that's why we say mix of oxygenated and deoxygenated. Definitely it's not like totally separate. It will have the mix. And you can see that it's oxygen poor, okay, except for pulmonary veins. Okay, so with this, I have done for the blood vessel.